Hello and welcome to the last section that we have under active transport. I'm trying to get to where we were at last. We ended off with endocytosis and we're going to finish this. It should be short, sweet, and to the point. Sounds like fun, right? Probably not, but we'll get it done. All right, here was the picture that I ended off with, the very last one. So this is video 3 of active transport and we got our 6 video notes done. So the next term that we have is exocytosis. And the exo means outside. So this is vesicle movement outside the cell. Normally, this is where the proteins that the cell has made is leaving the cell, or it's trying to get rid of waste products. And so it's kind of like the same idea. The vesicle fuses with the cell membrane. So on my picture here, I have on the bottom a vesicle that's a little bubble. And in the second part of the bubble, it's actually hitting that cell membrane, so it's going to fuse with it. In the third picture, it hits number 2, where that vesicle breaks open. And the last one, the molecules are out of the cell. I'm going to skip the video. We're actually going to see that in class, so we'll have some time to kind of explore this idea of exocytosis, materials leaving the cell. So it's just like endocytosis, but we're going to do it in reverse. Okay, there's also receptor protein vesicle movement, and this is where it's using that receptor-mediated endocytosis. And this is where we have a protein that it's going to bind to some sort of signal molecule. And that enables the cell to respond to some signals from the brain and nerve cells. So we're looking at a certain type of cells. When the signal molecule binds to a receiving cell, a change occurs. And like for some of these, as we're talking about like brain and nerve cells, that might be like a neurotransmitter, which can cause changes within your body. So like dopamine will make you feel good about yourself. And this allows these brain messages to be sent and received and for muscles to work. Sometimes some of these messages might be pain. And so that's where that pain receptor will receive it and it's going to spread to the next neuron and it's going to go out all the way up to brain and your brain's going to say, oh my gosh, this is really painful. Now, some of your medicine will try blocking some of these receptors so you don't feel the pain. And that's where then you might use like an ibuprofen or a Tylenol-based product to prevent then you getting that signal of pain. Novocaine is another one that works like when you're in the dentist's office, so you won't feel the pain of the dentist drilling. So there's a whole variety of different ways that we can use this, and these are going to cause a change within your body. So let's look at some of these changes that can happen with it. One of them is a permeability changing of the receiving cell, and that's going to cause those ion channels to open. Yeah, it might open that sodium-potassium pump and have all this sodium come flooding in, and now the cell's overwhelmed with all the sodium inside, and it doesn't want the sodium inside, so now it's going to have to use that sodium-potassium pump to remove sodium. It might trigger the formation of yet another messenger called a second messenger. And so that acts as a signal molecule and can increase a message or change to a cell function. And the last one is to activate some sort of enzymes in the cell. And then what that's going to do is either speed up the reactions or activate other enzymes. So here is a picture. Here is some ion gate channels. And here they're showing a neurotransmitter. So I kind of talked about a neurotransmitter. If you take anatomy, you're going to talk about a little bit more. So it binds to the protein. It's going to open that. Ions are going to flow across. And then what you can see is then in the second picture, same thing, neurotransmitter binds. It has a protein that's activated. And then that subunits actually control some of those ion channels. And so now you have an ion channel open and it's going to allow ions to move through. So here's kind of 2 different ways that you can see that happen. Okay, don't worry about these videos. We can see them later on in class. Yay! But this is my last part. Bum, bum, bum. You will need a full sheet of paper on this, and you will need to make a T-chart. you are going to put passive transport on the left and active transport on the right. You are going to put what you've learned so far about each type of transport. You'll need to include characteristics of each, substances that use each, mechanisms that each include, and information about each mechanism. So for example, if I say this one moves down the concentration gradient, you'll have to figure out, first of all, is that active or passive, and put it in. And you'll know that the other one then will move up the concentration gradient. Or if I say this one requires energy, you'll need to decide if that needs to go under active or passive. And then you'll need to find out then which one does not require energy. If I use the term osmosis, you'll have to figure out, is osmosis active or passive, and put it underneath the correct one. Then you need to explain which one works with osmosis. If I use the term sodium-potassium pump, which one uses the sodium-potassium pump? And then explain to me how the sodium-potassium pump works. If I use the term endocytosis, I'll have to go ahead and figure out which one uses endocytosis, active or passive, put it underneath that one. I'm going to explain how it works, and then I also probably will go into phagocytosis, pinocytosis, and receptor-mediated endocytosis. So you need each one of these underneath, and there's more that we have that I have not explained here. So go through your notes, and this is going to be a nice way to organize your notes. It's also going to be a nice set of cliff notes. So this is going to take you a little bit while, and this is then your last part of the notes. I expect to see this in your notes. You are graded on this section on your notes. If you do not have it, you will have a binder deduction come mid-quarter, which is right before Thanksgiving break. All right, if you have any questions on this, please ask me in class, or if you start working on it and you want to make sure it's looking okay, ask me as well, and I can go over stuff. But again, this is a nice way to kind of organize your stuff so you know what goes where and how things work. All right, you survived the last video. Hooray! And now you're going to be busy working on your binder in this T-chart. All right, have a great day.